GTP, and another prime conformation. We'll see examples of these conformational changes as we go. Okay, and so in its GTP bound form, this GTPase then can hydrolyze the GTP to GDP plus inorganic phosphate. So this is a hydrolysis reaction. So we use up water and we produce GTP and inorganic phosphate. Okay. This reaction often will not occur spontaneously. The GTPase needs to be activated. And so there are GTPase activating proteins, GAP, that will bind to the GTPase and allow it then to catalyze this reaction. Okay. And now, once we have the GDP, we need to release the GDP. Sometimes that will occur spontaneously, but with some of these GTPases, we need a guanosine nucleotide releasing factor, which triggers the release of GDP so that another GTP can bind. Okay, and we can repeat the cycle. Okay. So let's go through the three phases one by one. Okay. Let's talk about initiation. And before we talk about it, what happens during initiation, we need to talk about the initiation signal. Okay. And so in bacteria, the initiation signal consists of a start codon. Translation actually in all organisms always begins at an AUG codon. If you look at the codon table, AUG stands for methionine, and so every primary translation product begins with a methionine. Okay. In bacteria, this methionine is modified, okay, and so it's actually a formal methionine. It's a methionine that contains a formal group attached to the amino group. Okay. In addition to this AUG initiation codon or start codon, upstream, about 10 nucleotides upstream in the messenger RNA, we find something called a Shine-Dalgarno sequence. Okay. That is just a AG-rich sequence that has complementarity to a sequence at the 3' end of 16S ribosomal RNA. And so here we see a little fragment of a messenger RNA bound in the bound to the ribosome. And so remember, there's this groove between the head. You can't really see the groove very well here, but there's this groove between the head and the body of this 30S subunit, and the messenger RNA resides in that groove. Okay, and what we see here is that the start codon is bound. The start codon, this is the 5' end, and this is the this is the 5' end, and this is the 3' end. The start codon is now bound in the P site, right? And the second codon is bound in the A site, okay? And the Shine-Dalgarno sequence is actually making a duplex with the 3' end of the 16S ribosomal RNA. Actually, it's a little bit hard to see what's going on there, so I've found this structure in the protein data bank this morning, and here we can look at it in Pymol. Let me dim the lights. Okay. So here we're just looking at the 30S subunit of the bacterial ribosome for everything except for the 16S ribosomal RNA. And in addition, here's a Shine-Delgarno sequence, okay? And that Shine-Delgarno sequence shown below here, you can almost see it at this point, is actually forming a duplex with the 3' end of the 16S ribosomal RNA. And so if we go to this scene here, we can zoom in on that duplex. Okay, so here is the Shine-Delgarno sequence, and it's making a little RNA helix with the 3' end. So here's the very 3' end, and so this is the 3' end of the 16S ribosomal RNA. And then that positions the start codon in the P site. And as a result, the second codon is then positioned in the A site. Okay. All right. Okay. So that's how a start codon is recognized in bacteria. Actually, in eukaryotic cells, there's a completely different way of recognizing the start codon, and that's shown in this slide. So here is a eukaryotic messenger RNA. And remember, eukaryotic messenger RNAs all contain this 7-methyl-G PPP cap at the 5' end. And so what happens is that a eukaryotic initiation factor called EIF4E binds to this 7-methyl-G PPP cap, and then that recruits this 40S subunit, the small subunit of the eukaryotic ribosome, to the 5' end of the messenger RNA. And then this 40S subunit basically scans down the messenger RNA with help from RNA helicases, which melt out any stem loop structures in the 5' untranslated region, until the subunit encounters an AUG. Okay, once the AUG is encountered, now the 60S subunit binds and translation begins. Okay, and so this mode of recognition means that eukaryotic messenger RNAs are almost always monocistronic, right? So we know that bacterial messenger RNAs, they can be monocistronic, but they're often polycistronic. So a cistron, that's just an old-fashioned name for a gene, right? But genes in bacteria are often arranged into operons, and an operon then gives a messenger RNA. Okay, and so in the example we encountered of a polycistronic messenger RNA is the messenger RNA encoded by the lac operon, and that messenger RNA actually contains the coding information for 3 genes, 3 cistrons, right? There's LAC-Z, which encodes beta-galactosidase, LAC-Y, which encodes the lac permease, and then LAC-A, which encodes, I think it's called a thiogalactoside transacetylase. Okay, and so we have a polycistronic messenger RNA. Each one of these cistrons, each one of these coding regions then begins with an AUG start codon. And then just upstream of each AUG start codon, there's a Shine-Delgarno sequence, an SD, a Shine-Delgarno sequence. Okay? And so the ribosome can bind independently to each of these Shine-Delgarno sequences, and that way a single message can give rise to multiple proteins. We can have polycistronic messenger RNAs, but in eukaryotes, we cannot have, for the most part, polycistronic messenger RNAs, And that's because the way the ribosome finds the AUG is just by binding to the 5' end, and then it always initiates translation at the first AUG it encounters as it scans down the messenger RNA in the 5' to 3' direction. Okay. Okay, so in addition to a start codon to initiate translation, we need an initiator tRNA. Okay, and so the initiation codon is always a methionine codon, and so the initiator tRNA is a tRNA methionine. Okay, and it turns out that in both bacteria and eukaryotes there are two tRNA methionines. There's one that's responsible for recognizing initiation codons, and there's another one that's responsible for recognizing internal methionine codons. You know, we have methionine at the beginning of each translation product, but we also sometimes have methionines internal to proteins, right? Methionine can appear anywhere in a protein, not always just at the first amino acid, right? And so here we see what happens in E. coli. There's two tRNAs. There's tRNA F-met, okay, which is the initiator tRNA, and then there's the tRNA M-met, which is responsible for reading internal methionine codons, internal AUG codons. Okay, both of these are charged by the same methional tRNA synthetase, okay, to give us methional tRNA F-met or methional tRNA M-met. And then this one, methional tRNA F-met, it actually gets formulated by a transformulase that takes a formal group from N10 formal THF and transfers it onto the amino group of the methionine attached to the tRNA F-met. And so now we have formal methional tRNA F-met. Okay, so why do we need these two different tRNAs, one for reading initiation codons and one for reading internal AUG codons? Well, it's because they are recognized by different translation factors. Okay, and so this particular tRNA species is recognized by this initiation factor, IF2, which delivers it to the 30S subunit before translation begins. And this particular tRNA is recognized by the elongation factor, EFTU, which then delivers it to the ribosome during the elongation phase. Yes, so.
Is that a small typo up there? Because for like bacteria and eukaryotes, I think you meant like both have tRNA F.
No, in eukaryotes, since it does, so the F stands for formal, okay? So because we end up getting this formal methional tRNA F-met. In eukaryotes, the initiator tRNA goes by a different name. It's called tRNA I-met because the methionine does not get formulated. So we don't have formal methionine at the beginning of translation products in eukaryotes, just plain old methionine. And in this case, the I stands for initiator. It's kind of fairly idiosyncratic nomenclature. Was that your question, Osan, or am I answering the wrong question?
Oh yes, that, and also just to clarify, like eukaryotes still need to formulate the methionine tRNA complex to make it distinguishable as a start codon recognizer.
Well, I think the reason we have this formal group here is probably to protect the N-terminal end of the polypeptide from proteolytic degradation. Why we don't need that in eukaryotes is not clear. There must be other ways of protecting the N-terminal ends of primary translation products from proteolytic degradation. I don't think it's strictly required for initiation. It's just there. It protects the N-terminal end of the primary translation product from proteolytic degradation. Okay? All right. Okay, so here is a view of the initiation sequence in E. coli, and we're going to look at this again in an animation in a few minutes. Okay, so we have the 30S subunit. It binds to this initiation factor called IF3, which prevents the premature reassociation of the 30S subunit with the 50S subunit. And now it binds more or less all at once to everything else. It binds now to the initiator tRNA, the formal methional tRNA FMET, okay, and it binds to the messenger RNA, which contains the Shine-Delgarno sequence, and binds therefore to a sequence near the 3' end of the ribosomal RNA. The ribosomal RNA is not shown here. Okay, and so now we form this so-called 30S initiation complex in which the initiator tRNA is bound in the P site, in basically what's going to become the P site, and it's bound in part to this codon-anticodon duplex, right? We have the anticodon loop here, which is complementary to the AUG start codon. Okay. At this stage, the tRNA, the initiator tRNA, is bound to this initiation factor, IF2, which is one of those GTPs. The IF2 only has affinity for the tRNA when it's in its GTP-bound form. Okay. So that we call this the 30S initiation complex. Now the 50S subunit comes along and binds to the 30S initiation complex to form the 70S initiation complex. When that happens, it displaces IF1 and IF3, and it triggers, it acts as a, I think it says that here, it acts as a GTPase activating protein to trigger hydrolysis of GTP to GDP. That causes a conformational change in IF2, which allows it to let go of the tRNA. And so as a result, IF2 is also released now in its GDP form. Okay. In order to recycle the IF2, it needs to release the GDP so that it can bind another GTP and then associate with another molecule of initiator RNA. In E. coli, that GDP release occurs just spontaneously. It's a little bit slow, but it occurs more or less spontaneously. You don't need a guanosine nucleotide releasing factor. We're going to see that in eukaryotes, we do need a guanosine nucleotide releasing factor at this stage to release the GDP, and that actually plays a role in the regulation of translation. Okay, so keep that in mind. We probably won't get to that today. We'll talk about it on Monday. Okay, so now we have a tRNA bound in the P site. The A site is empty. Now we need to begin elongation. Okay. And so the first step in elongation is we need to bind an amino acyl tRNA in the A site. So here's the amino acyl tRNA. It forms a complex with EFTU, which is this GTPase. And the EFTU essentially delivers the amino acyl tRNA to the A site. Once it does that, we need to get rid of the EFTU, and so it turns out that this ribosome then acts as a GAP, a GTPase activating protein. As a result, the EFTU hydrolyzes the GTP, turning it into GDP. It then loses affinity for the amino acyl tRNA, and then so that it can be released from the A site. Okay, and now it turns out we need a guanosine nucleotide releasing factor, which is one of the other elongation factors called EFTS, to stimulate the release of GDP so that we can bind GTP and repeat the cycle. Okay, so now an amino acyl tRNA has been delivered to the A site. Transpeptidation can occur. The peptide in the P site can be transferred onto the amino acid on the amino acyl tRNA in the A site. In the process, we've converted this now into a peptidyl tRNA. It was an amino acyl tRNA, now it's a peptidyl tRNA, and we've elongated the polypeptide by one amino acid. Okay. And we've converted this tRNA into an uncharged tRNA. Okay. All right, so what happens next? Well, now translocation needs to occur. Okay, the whole ribosome needs to move down the messenger RNA by 3 nucleotides, by 1 codon. Okay, so translocation, movement of the ribosome along the messenger RNA needs to occur, and that is facilitated by another GTPase called EFG. EFG binds to the ribosome in its GTP-bound form and then hydrolyzes GTP. That induces a conformational change which pushes the ribosome down the messenger RNA. Okay? In the process, then, the peptidyl tRNA moves into the P site. The uncharged tRNA moves into the E site, which isn't shown here, and then it's released from the E site into the surrounding media. Okay? And so now we're back here where we started, and we can do the cycle over and over again, adding as many amino acids as we need before we encounter a stop codon. So this is another view of the elongation cycle, which shows some details that are left out of that previous slide. And so now we're actually showing the E site. Okay, so here we are. Okay, so that picture here is equivalent to this picture here, except we're showing things in a much more schematic way. And so in a way, there's less detail, but in a way, there's more detail, as you'll see in a minute. Because, for example, we are showing the E site now. We have an uncharged tRNA in the E site. We have a peptidyl tRNA in the P site. And the A site is empty at this stage. Okay. And so now we have to deliver an amino acyl tRNA to the A site. So here we have an amino acyl tRNA bound to EFTU in its GTP bound form. The job of the EFTU is to deliver the amino acyl tRNA to the A site. Okay. But that delivery occurs in 2 steps. And so those 2 steps were telescoped into 1 step in the previous slide. So initially what happens is that the anticodon stem loop binds to the 30S subunit in the so-called decoding site, okay? And so it binds then to the codon in the 30S subunit, but the acceptor stem cannot enter transferase active site in the 50S subunit. And the reason it can't do that is because it's still bound to EFTU, and so EFTU is too big to fit into the A site. Okay? And so then EFTU hydrolyzes GTP. That causes a conformational change, which allows it to release from the amino acyl tRNA, and now the acceptor stem can spring into the A site. Okay, so this is called a hybrid state because the anticodon stem loop is in the A site, but the acceptor stem is not yet in the A site, and so we call this the AT hybrid state. So it refers to the fact that down here we're in the A site, up here we're still bound to EFTU. So I guess the T stands for EFTU. Okay? Okay, so now we're ready for transpeptidation. Okay, and so the transpeptidation reaction occurs. We've looked at that transpeptidation reaction in quite a bit of detail. We know, something about how it's catalyzed by the ribosome, presumably acting as an entropy trap. Okay. All right. And now what happens? Well, now we need to move everything into the adjacent tRNA binding site. Translocation needs to occur. But translocation actually also occurs in two steps. The first thing that happens is that the acceptor stems move over one tRNA binding site. And so now that this tRNA is uncharged, it will get into the E site. Its acceptor stem will fit into the E site. And so this acceptor stem moves into the E site, and the acceptor stem of the new peptidyl tRNA moves into the P site. And so we have two hybrid states here. We have the PE hybrid state for the uncharged tRNA, and we have the AP hybrid state for the peptidyl tRNA. Okay. And now, so this is called the pre-translocation state because the ribosome hasn't yet moved down the messenger RNA. And now with the help of EFG, the ribosome moves down the messenger RNA, and as a result, the anticodon stem loops also move over into the adjacent sites. And so now we're back to this post-translocation state. Okay. All right. So let's look at the movie, and then it'll be time for the quiz. Yeah. Yeah, Connor, you can go ahead and pass out the quiz, but don't start working on it. And please pay attention to me. All right? I need to rewind the video here. Okay. Please quiet down. All right. So here is an animation showing the entire translation cycle in E. coli. Okay, based on crystal structures made in Ramakrishnan's lab, and then this beautiful animation was also made in Ramakrishnan's lab, one of those Nobel laureates I told you about last time. Okay, all right. Okay, so I'm going to stop the video and just advance through it more slowly using the arrow keys here. Okay. Yeah, I have 10 minutes because we don't need to start until 10:40 because you have 10 minutes to, then that'll give you to do the quiz. Yeah, please. Okay. So what do we see here? We see the 30S subunit. We dim the lights. We see the 30S subunit, and we see the messenger RNA. They're flying towards each other. Okay. And also now coming into view is in blue here is the aminoacyl, the initiator aminoacyl tRNA, the formal methional tRNA, FMET. So the blue here is the tRNA, and you can see the little formal methionine bound here at the tip of the acceptor stem. Okay. And now everything binds all at once in that groove between the head and the body. Okay. Okay, and now we have the 30S initiation complex with the 30S subunit shown here in this, I don't know what color that is, pinkish, tannish color, the formal methional tRNA, FMET, bound to the messenger RNA. Here's the messenger RNA, which we can barely see. Okay. And here is the anticodon stem bound to IF2. Okay. Okay. Need to start it again. Okay, there it's good. It pauses at that shot for quite a while. Okay. And so now what are we doing? Well, now we need to bring in the large subunit. Here comes the large subunit. Okay. The large subunit binds. It displaces IF1 and IF3, which are those red and green blobs there that are now floating away. The orange blob IF2 is still bound. The large subunit then triggers hydrolysis of GTP. Okay, and now IF2 can dissociate. And so now we have the 70S initiation complex. Okay. Okay. And so now we can begin elongation. Okay, so now what's happening? Now the aminoacyl tRNA that is the cognate aminoacyl tRNA for the second codon in the messenger RNA comes in. It comes in as a part of a complex with EFTU. So here's the aminoacyl tRNA in purple. Here is EFTU, this whitish blob here. Every time I stop, I have to restart. Okay. Oh, what just happened there? Yes. So you see that flash of light? That flash of light signifies that EFTU has just hydrolyzed GTP, right? There's no flash of light in reality, but that's their way of signifying the hydrolysis of GTP. Okay. And now the EFTU undergoes a conformational change and it's released, and now the acceptor stem can swing into the A site. Okay. And now what just happened there? You saw this kind of like morphing of the surface of that purple tRNA. See how it's doing that? I think what that signifies is the transpeptidation reaction. We can't see the peptidyl transferase active site because it's buried in the 50S subunit, but when that morphing of that purple surface occurred, that's telling us now that the peptidyl tRNA, that the peptide is being transferred from the blue tRNA onto the purple tRNA. Okay, so now the peptidyl transferase reaction has occurred. Okay. All right. And now what's going to happen? We need to get those hybrid states forming. We need those acceptor stems to swing into the adjacent sites. And now EFG comes along. EFG, we're going to see another flash of light. There it was, hydrolyzes GTP, and now undergoes a conformational change which allows it to push the messenger RNA through the ribosome. So now translocation has occurred, okay? And now the next amino acyl tRNA is going to come in and bind in the now unoccupied A site. Okay, here it comes, bound to EFTU. Okay, all right. For a while there, they took away part of the 30S subunit to make it easier to see what was going on. Now they brought it back. Okay. And when the new amino acyl tRNA binds, that's actually the signal for the uncharged tRNA to leave. You can see the blue uncharged tRNA leaving. Actually, they repeat a little bit of this for reasons that are not that important. Okay, so let's just look at this at regular speed now. And so we can see one amino acyl tRNA coming in after another, elongating the polypeptide chain. The polypeptide actually exits from the ribosome through a pore that runs all the way through the large subunit, and here it is emerging from the backside of the ribosome. As it does so, it assumes its tertiary structure. Okay. And then finally, we're ready for termination. Termination occurs when, okay, let's back up a little. Termination occurs when a stop codon comes into the A site. There's no amino acyl tRNA that can recognize a stop codon. So instead, this protein RF2 binds in the A site. And what does it do? It reaches up there and it triggers the hydrolysis of the carboxyester linkage so that now the polypeptide is released. So you can see now the polypeptide has been released. Okay. And then another release factor comes in, it's not shown here, and it induces the release, we'll speed things up again, induces the release of RF2. So RF3 comes in and induces the release of RF2, and now this so-called ribosome releasing factor comes in. EFG makes a repeat appearance and pushes the final tRNA out, and then the two subunits associate with one another, and we are ready to translate yet another messenger RNA. Okay, so yeah, put everything away except your pencil or black pen and go ahead and do the quiz. Let me turn the lights on so you can see what you're doing.